Hi everyone, my name is Kristen McGennick.
I'm Nicole Hickman.
I'm Ellery Fitzgerald.
I'm Gabriella Monkowitz.
And our presentation is going to be on figure I through L of human iPS cell-derived dopaminergic neurons functioning in a primate's Parkinson's disease model. So this molecule is dopamine. It looks relatively simple. However, it is going to be the focus or one of the focuses of our presentation today because it is very important in Parkinson's disease, or the lack of the molecule is important because the lack of it will produce the symptoms that we see in Parkinson's patients.
The objective of this study is to see if iPS cells can treat Parkinson's disease and give more dopamine levels to the brain than the drug L-Dopa. Our research question is, over the course of a 21-month duration, scientists observed the progress of the grafts that were implanted into monkeys' brains. Do the images as well as the data suggest that the dopaminergic neurons produce enough dopamine so that the symptoms of Parkinson's disease will be suppressed? The study is focused on the interactions of the structure of the brain. Within the brain, there is the putamen, which is where it interacts with structures and controls voluntary movement. In addition to movement, the putamen will affect different parts of learning and memory. L-Dopa drug is used for treating Parkinson's disease, but scientists are trying to figure out whether human iPS cells are going to be more effective for treating the disease. Having a test subject as a primate with similar genetic material as humans would produce similar results for testing the grafts of the dopaminergic neurons from iPS cells in humans. By injecting monkeys with MPTP and inducing Parkinson's disease, we can test the efficiency of iPS cells. Using MRI graphs and PET scans, we are able to look at the dopamine and activity in the neurons over a long period of time to see if the differentiated dopaminergic neurons. To test this treatment, we can place iPS cells in a petri dish and let it grow, and once it is ready to transport into a subject for testing, then we can graft the cells into the brain tissue. The objective of the research report is to analyze the data given to us for the neuron activity in the cells of a monkey's brain. The result wanted is that human iPS cells will be more effective than the L-Dopa drug, and by comparing the long-term effects of both tests, there will be a better understanding of treating PD patients. The people conducting the experiment will be looking for the results of the cell grafting over a 21-month duration. By injecting monkeys with MPTP and giving them symptoms of Parkinson's disease, we can test the efficiency of iPS cells. To test the treatment, we can place human iPS cells in a petri dish and let it grow, and once it is ready to transport, graft the cells into the brain tissue of the subject.
So this is the experimental workflow. We're starting with the fibroblast cells of 8 patients, some of which are healthy and do not have Parkinson's, and the others are unhealthy and unfortunately do have Parkinson's. Once you get the fibroblast cells, they culture them in a dish with a cocktail that forces them to basically convert back to pluripotent stem cells, and they will then differentiate into dopaminergic proliferation or proliferating neurons. Once they get to the proliferating stage, they will be grafted into the brains of this monkey. And once it's grafted in there, they will wait a 21-week or 21-month period of time. And during these weeks and months, they will be injected with something called fluorodopa, which is a radioactive tracer molecule. And this molecule allows us to use things like PET scans. So it'll light up the brain and show us where the dopamine as well as where the dopaminergic neurons are. And once we get these images, as well as give them a couple of more tests, we can turn our data into graphs, and that's what we'll be looking at a little bit later.
So this is the MRI scan from the research report. So MRI stands for magnetic resonance imaging, and it uses magnetic forces to align protons with a magnetic field to capture images of tissue. The graph in image I shows the MRI scan of a monkey's brain that received PD and the treatment. The putamen is represented by the dotted lines in the image created by the brain, and this is where the activity levels will be monitored in a PET scan. We can view the cerebral fluid with the black circles surrounding the dotted lines, showing where the putamen is. To check the graph's volume, we look at the gray and white matter and cerebral spinal fluid to make sure the brain is working properly.
So this is positron emission tomography, or a PET scan. And in this image, or in this imaging technique, they will inject a patient with radioactive tracer dye. And once they inject them, it will go to areas of the body that there's proliferating cells. And it's usually used for cancer because cancer cells will proliferate and divide more quickly. So it's easier to see cells like this, but it can also be used to trace certain chemicals that they have pinpointed them to. In this image, we can see that there's these little blue lights going on and blue glow. And this one we see what an SUV, which is a standard uptake value. This is basically the chart that tells us how much of the dopamine and the dopaminergic neurons have actually been implanted and how much they have grown and spread over time. So as we can see, this one is a little bit blue, which means it'll be about right here. And this is probably an earlier photo because there's so little neurons that have been proliferated inside the brain. But later on, we'll see that it gets a little bit redder.
So in this experiment, the scientists took MRI scans and examined dopamine synthesis using F18 DOPA PET scans. In the MRI scans, they created T1 weighted images divided by the T2 weighted images. This automatically segmented the brain into 4 parts: gray matter, white matter, cerebral spinal fluid, and graft. These MRI scans were to check the graft's volume. The white matter is the lighter regions of the image, while the gray matter is the darker parts. The graft would be the circled area. The cerebral spinal fluid is the darker area towards the middle of the MRI image. The scientists found an increase of F18 DOPA in the putamen. The F18 DOPA PET is a radioactive tracer for PET, which can visualize the nerve endings of dopaminergic neurons. For figure I, the white lines in the image shows the putamen. The C11PE2I is a radio tracer in PET targeting the neuronal dopamine transporters. In the F18 DOPA CRB ratio and the C11PE2I BPND, it has a color scale from 1 to 2 and 0 to 3. In the DOPA and PE2I images, the light blue areas towards 1 and 0 show lower activity levels, and the colors going closer to red, closer to 2 and 3, mean that the area is more active. In the images, you can see the area circled is becoming more red when getting closer to 21 months, which shows a higher activity level.
So for part I, we can conclude that the MRI and the PET of the monkey's putamen is showing increased activity of the dopaminergic neurons because the pictures are increasingly becoming more red over time, thus meaning the dopaminergic neurons are actually functioning and producing the dopamine over time.
In graphs J and K are the Ki values recorded from the F18 DOPA PET scan. In both of the graphs, it shows the Ki value versus the amount of time after transplant in months. As you can see in both graphs, there is an overall upward trend, and the dotted line shows the designated average for 2 intact monkeys. The constant Ki of the normal monkeys, the monkeys treated with MPTP and the ones with transplanted cells, showed that the monkeys with transplanted cells recovered to 48% of the normal monkeys. MPTP is a prodrug that causes permanent symptoms of Parkinson's disease.
So to conclude for parts J and K, as already explained, the Ki values and the amount of time after transplantation are graphed. The time after transplantation is graphed on the x-axis in months, and the Ki values are graphed on the y-axis in 1 × 10⁻³ per minute. And the lines show the mean values n equals 3 for the vehicle and PD groups and n equals 4 for the healthy group. These lines indicate that the transplantation was restoring the NPTP treated monkeys' dopamine levels by at least 48% compared to the normal monkeys' levels.
So for graph L, it shows the Ki value versus the number of surviving TH plus cells found. The healthy group of monkeys is shown by the blue dots, and the PD group of monkeys is shown by the red dots. The graph has an upward trend line showing that there is a significant correlation between the Ki values and the surviving TH plus cells in the graphs. This suggests that the F18 DOPA PET is able to monitor the survival of dopaminergic neurons.
For part L, the graph shows the number of TH plus cells times 10 to the 5th power versus the Ki values 1 times 10 to the negative 3 per minute. The upward linear regression line and the correlation value suggest that the survival of the dopaminergic neurons can be monitored by the DOPA PET test. Therefore, since the dopaminergic neurons are surviving in the MPTP treated monkeys, the treatment is working in reducing the degeneration of these neurons, leading to a decrease in Parkinson's symptoms.
So to kind of just summarize all of those conclusions for each part of our figure, we have part I. So because the red in the spectrum indicates the level of activity of the dopaminergic neurons in the putamen, and since the MRI and PET photos are becoming increasingly red over time, this means that the levels of activity of dopaminergic neurons are progressively increasing over time. Overall, this indicates that the dopaminergic neurons are progressively producing more dopamine in the putamen over time, and the researchers' experiment of transplanting the progenitor cells into the MPTP-treated monkeys is helping the dopaminergic neurons to stop degenerating and therefore decreasing the symptoms of Parkinson's disease. And for part K and J, they also show positive results for the researchers because the Ki values versus the time after transplantation show an increase in the MPTP-treated monkeys' dopamine levels. The dopamine levels in the NPTP-treated monkeys were restored by 48% compared to the normal monkeys' levels, also indicating that the transplant of the progenitor cells is helping to stop the degeneration of the dopaminergic neurons and therefore decreasing Parkinson's disease symptoms. And then lastly, for part L, the positive correlation between the TH plus cells and the Ki values represent the survival of the dopaminergic neurons can be determined by DOPA PET. The survival of these dopaminergic neurons shows that the progenitor cell transplant is working yet again to stop the degeneration of the dopaminergic neurons, as already stated, and therefore decreasing the symptoms of PD.
This letter could use better explanations. For example, the Ki value and the vehicle wasn't talked about in depth, making the experiment difficult to understand. There needs to be an explanation of what DAT is and why it is used in the experiment. For the color bar, there wasn't a clarification of what the colors mean with regards to the study. This makes the interpretation of the PET scan difficult. In figure I, there is a blank square where it looks like an image wasn't taken. The letter does not discuss why there isn't an image for the F18 DOPA scan for week 1. There also needs to be a better explanation of the reasons they took the MRI scans. What is its purpose in the experiment?
These are all our sources for the images and some of the extra information that we weren't really sure about. Thank you for watching.
Thank you.